Okay, so we are going to model the structure of organelles in eukaryotic cells. Some of the organelles we're going to model are going to be the nucleus, the plasma membrane, the cell wall, mitochondria, vacuoles, chloroplasts, and ribosomes. Okay, so the first part of the cell organelles that we're going to model is going to be the cell wall and the plasma membrane. So the way we're going to do this is we're going to go up here to the sketch tool. We're going to move the mouse over to the spline where it says spline, create a spline, and we're going to left click. And then as you can see, if you move your mouse down, you get a dialog that says click on grid. And if you click anywhere on this grid, you can see it extends your grid out, and you get a crosshair on underneath your mouse. So what we're going to do is we're going to trace out our cell wall with this tool. So if you left click and then move your mouse, you can see you can start tracing out the shape you want. So you just keep left clicking until you get a general shape like you want. So we're just going to do a general shape of a cell. So just going to, doesn't have to be crazy elaborate. Just keep left clicking. Until you get all the way back around to where you started. And once you connect that last point, it should look something like this. It should fill in with a color. And then what we're going to do is we're going to go to the construct tool. We're going to go down to the extrude tool and left click. And as you can see, if we click the design we made, you get an arrow and a little number over here in millimeters. So what we're going to do is we're going to drag this left click and drag this arrow upwards like this. And as you can see, once you let go of the left mouse button and click away, you get a 3D model of the shape you traced out. The next step to do this is we're going to click our shape, and we're going to do the scale tool over here. So if you go to transform and then go to scale, you can scale this however to whatever size you need. So we're just going to scale it down a little bit, and we can delete away our initial spline design by clicking it and pressing the delete button on the keyboard. So what we're going to do next is we're going to make a copy of this shape we made. So we're going to click it, and we're going to press Control C on the keyboard, and then we're going to press Control V as in violin, and it'll make a duplicate of the shape. And then we're going to go over here to the transform tool. We're going to go back to scale, and we're going to take this arrow and drag it to the left just a little bit, just about that much, and let go. And what that does is it creates a smaller object inside our larger design. So now we're going to click our larger design, and we're going to click the top, so only the top is highlighted like this. You can see the outline like this. And we're going to click this little gear that shows up, and we're going to go to the third tool, which is called shell. And what this will do, if we click it, left mouse click it, it will hollow out everything but the edge of our design. As you can see, our slightly smaller, scaled down version is still in there. And what we can do with this is we can click it, click the design in the middle, click material, and we click frosted glass, and then we click the close out of this window. Like this. You just make sure you double click it and close out of this window. And as you can see, it makes a pretty clear design, like a cytoplasm inside our shape. So this is a pretty simple design of a cell wall. But something that's right inside the cell wall is the plasma membrane. So what we're going to do is we're going to select the outer design. We're going to copy it again and paste it again with using Control C and Control V. And we're going to scale it again by going to Transform and Scale. And we're going to drag the Scale tool arrow right here in the middle towards the left just a little bit. And let go. And after we do that, we're going to click Material, and we're gonna choose a different color just so we can differentiate between the two. So it can be any color in this using this color wheel. So you just click a color, and we can close this window. And as you can see, now we have a cell wall, a plasma membrane, and a cytoplasm inside. Okay, so the next organelle that we're going to model is the nucleus of the cell. And we're going to do this by scrolling out a little bit using the scroll wheel so we can get a better view. And we're going to go to the primitives tool, and we're going to select a sphere. And we're going to drag it over here in just an open area and left click. We're going to try to just focus on that. So the next thing we're going to have to do is scale this up a little bit using the scale tool. So we go to Transform, and then we go to Scale. And we can use this arrow to scale what's going to be our nucleus up a little bit so you can see it better. The next thing we're going to need is we're going to need to go back to primitives, and we're going to have to select box and just put it right beside our sphere. And we're going to have to scale this box just like we did our sphere, but we're going to have to do it in a specific way. What we're going to do is we're going to go down to this tool where it says Scale Uniform. We're going to click the arrow right beside it, and we're going to go to Non-Uniform. And as you can see, when you do that, you get 3 arrows instead of 1. So what we're going to do is we're going to look down on our box from the top and make sure that it's longer than our sphere. And we're going to look at our box from the side. Make sure you have scale still selected, non-uniform. And we're going to make sure it's taller than our sphere. And then we're going to just drag it out a little bit to make it a little wider. And we're going to go back to the Transform tool. We're going to go to Move and Rotate tool. And using this arrow right here, we're going to drag the box to the left toward our sphere. So it covers up half of the sphere. Just right about half, like this. And then we're going to use the up arrow, and we're going to move that halfway up, so half of the box is covering half of the sphere. Now what we're going to do is we're going to go to Combine, and then we're going to go to subtract. And then we left click the sphere, and then after that we left click our box and we press enter on the keyboard, and as you can see it etches out wherever the box was on the sphere. So the next thing we're going to have to do is put a sphere in the center of this, right about there, and we can move it using the move rotate tool to make sure it's in the center. And we can change the colors of this by going to the material setting and just picking a color. So we'll pick a color for that, and we'll pick a color for the outside. Let's do this color. Okay. So now we're going to click and drag so we highlight both of the spheres, and we're going to go to the move and rotate tool, and we're going to drag our new nucleus into the cell. Okay, now that we have our cell wall, our plasma membrane, and our nucleus, the next thing we're going to model is the mitochondria. Now, we're going to model our mitochondria to look a little something like this. So to do this, we're going to go to the primitives tool, we're going to go to sphere, and we're going to put it over here just like we did when we made our nucleus. Now this time we're going to go to our scale tool, and we're going to go to non-uniform again, and we're going to elongate this sphere so it's more elliptical. And then once we have our sphere elongated like this, we're going to do something very similar to what we did with our nucleus. By going to primitives, we're going to select box and putting it beside what's going to be our mitochondria. And we're going to scale this box similarly to like we did when we made our nucleus by going to non-uniform again and making sure it's longer and wider than our elongated sphere. We're going to then move, using the move and rotate tool, this box so it covers the sphere, and we're going to drag it up so it only covers the top half of this. So something like this. And then we're going to go back to the combine tool. We're going to click subtract again, and we're going to click our elongated sphere first, and then we're going to click our box, and then we're going to press enter. And as you can see, it etched out wherever the box was, just like before. So now what we're going to do is we're going to select this, and we're going to click again to select the top. So as you can see, you can select the different parts, and we want to make sure we have this top part selected. We're going to click this gear, and we're going to click shell. And as you can see, it empties out or hollows out the middle. Just like this. So now what we're going to do is we're going to make the internal structure of this mitochondria, this right here, using splines like we did when we made the cell wall and the plasma membrane. So we're going to go back to the sketch tool, we're going to go to splines, we're going to click on the grid, and we're going to make a design similar to this inside. So what we're going to do is we're going to start by left clicking, right about here, and we're just going to trace out a design of the internal structure of that mitochondria. And then we're going to close the design, and it should look something similar to this. And then we're going to go back to the construct tool and click extrude and select this, and we're going to use this arrow to raise our design up. It should look something like this. So now what we're going to do is we're going to shell this design just like we did with the sphere. So we're going to make sure we have the top selected, we're going to click the gear, and we're going to press shell. This is what it should look like right about now. Just to make this a little easier, we're going to change the color of this by making sure you have it selected and going to the materials tool and picking a color. So now we're going to select the internal structure of the mitochondria. We're going to go to the move and rotate tool, and we're going to drag it inside our mitochondria. You're going to have to drag it inside, and then we're going to have to drag it up a little bit. So it should look something similar to this. Now we can click our spline and press delete on the keyboard, and we're going to highlight both of these parts by left clicking and dragging a box around these, and we're going to go to transform and move and rotate to drag it inside of our cell using the arrows. Just like this. Okay, now we are going to model a vacuole. Now a vacuole is going to look something like this green part right here. So to make this, we're going to go up here to the primitives tool. We're going to select a sphere and place it over here again. We're going to focus on it. What we're going to do is we're going to put a box right beside it again. We're going to scale it just like we did before using non-uniform. Make sure it's longer. and wider than our sphere. And using the move and rotate tool, we're going to cover half of this sphere with our box. Something like this. We're going to go back to the combine tool, and then click subtract. We're going to click the sphere first, and then we're going to click the box, and then we're going to press enter. You should end up with something like this. We're going to select the top of this. Make sure you have the outline of the top selected like this. You're going to click the gear, and you're going to press shell. And it should hollow out the center, something like this. And then you can pick a color for it. So we're going to color it this color. Oops. There we go. And now we're just going to use the move and rotate tool to move our new vacuole into our cell. Okay, now that we have our vacuole in our cell, what we're going to do is we're going to model a chloroplast. Now, this is what a picture of a chloroplast looks like. And as you might tell, it has a very similar shape to a mitochondria, but has different internal structures. So, the initial making of this chloroplast will be almost exactly like making the mitochondria. We're going to go to primitives, and we're going to click sphere, and we're going to scale this sphere non-uniformly, and we're going to elongate it again. So it should look something like this. We're going to go back to box under primitives. We're going to non-uniformly scale this again so it's longer and wider than our elongated sphere. And we're going to cover the top half of our sphere, just like we did before. Then we're going to go to combine, subtract, select the sphere first, and then press the box. Press enter on the keyboard. So you should end up with something like this. We're going to select the top of our elongated sphere. We're going to click shell. And instead of designing something like the mitochondria, what we're going to use, instead of using splines, we're going to use cylinders to simulate these structures inside. So what we're going to do is we're going to go up here to primitives, we're going to go to cylinders, and we're going to drag one over here. And we're going to scale this non-uniformly again, making it shorter and skinnier. So it's about proportional to our elongated sphere. Make it a little bit smaller. now what we're going to do is we're going to select this cylinder. We're going to press Control C and press Control V. And you can see it makes a duplicate, and this should show up these arrows, these move and rotate arrows, and we're going to drag it inside of our chloroplast. Like so. We're going to do this a couple more times, so we're going to select this cylinder over here. We're going to press Control C and Control V. We're going to drag this in, lower it down, and we're going to keep doing this until we have a couple inside of our new chloroplast. I'm going to drag this one forward. So that should be about enough. And we're going to drag this last one using the move and rotate tool into its position inside our chloroplast. Just like before, we're going to drag, using the left mouse button, a box around this so they're all selected. Using the move and rotate tool, we're going to drag this over into our cell. We're also going to change the color of this. Since it's a chloroplast, we'll do a nice bright green color. The final cell organelles that we're going to model are ribosomes. And the way we're going to model ribosomes is just using a very simple sphere. So we're going to drag it over. We're going to scale it down a little bit using the scale tool. We're going to change the color. And we're going to make a couple copies of this by pressing Control C and Control V and dragging them into our cell. Whoops. So you're just going to do Control C and then Control V. Until you have a few ribosomes inside your cell. And then we can just drag this last one using the move and rotate tool into our cell. Oops. And we have now modeled the cell wall, the cell membrane, mitochondria, a nucleus, chloroplasts, vacuoles, and ribosomes.